conclude what we believe to be the future direction of this. So, but apart from symptoms, we believe that if we manage to model this and understand these other phenotypes. So phenotypes, if you take a relaxed definition, you would think of it as a observable trait of the mental disorder, not necessarily a symptom that, or not at all a symptom that really bothers the person, but something that can be, for example, seen in behavioral or electrophysiology tests. And we have discussed this in more detail in this review from 2019. And these EEG-based phenotypes include, for example, altered spectral power. So there's increased or decreased delta oscillation power in different brain areas of schizophrenia patients, and also gamma and probably other frequency ranges as well. And then there are disturbances of these auditory event-related potentials and their derivatives, and also visual event-related potentials. I'll go a little bit in detail to this phenotype. So this is a phenomenon that where you give a checkerboard visual stimulus to a person and keep reversing the white to black and black to white for many minutes. Then the event-related potential that is related to each of those changes gets strengthened in amplitude by time. But how much it gets strengthened, is different between schizophrenia patients and healthy controls. And what I want to kind of argue is that it is becoming possible through both advances in computational modeling and genetic data to really link these phenotypes or map them down to a gene level. So starting from integrated and fire model, so it was introduced in 1907, so already almost 120 years old model, but still usable in some cases. So here you have only one variable per neuron to describe the dynamics. In Hodgkin-Huxley model, you have 4 different variables: the membrane potential, and then activation variables of the sodium and potassium channels. And so the beauty of this of the 70 years old model was that they could fit these model parameters into the electrophysiological data they measured from this quick action. And when we go to modern biophysical detailed model, so this is pretty much still the same Hodgkin-Huxley model. We just have the whole neuron morphology there instead of just single points. And we have, instead of just one sodium and one potassium channel, we have, for example, in this 2011 paper that we have used a lot, there are 2 sodium channels and 5 or 6 potassium channels and 2 calcium channels. And then also this axial current that is needed for this one. And when they fit this model, they fit this conductances of all these channels in different compartments alongside with these calcium dynamics parameters. And of course, this model is only as good as the error functions that you introduce for fitting this model, and that is where the challenge of making these models more suited to this kind of study lie in the future. Here you can see that if they fit this model to only perisomatic firing features, they get mildly different ion channel conductances and other parameters than when they only fit it to this backfiring. I won't go much into detail in this, but as you know, probably most of you know that then the beauty is that you can stimulate this neuron wherever and record it wherever you want and study the neuron response in different scenarios. And to go from a single neuron to networks, all you need is this synapses. Typically, people use chemical synapses, AMPA, NMDA, and GABA. And in this 2015 paper, they used this very same A model neuron and linked 150 of them together, and each of them they activated with background synaptic inputs, and then they also have this AMBA and NMDA mediated inputs within the network. I should also mention, so inspired by David's talk and other talks I've heard here, so we have also previously studied how the structure shapes the dynamics in this. So here we used point neuron models. But what we did was to generate different types of network topologies and then tested which features of these networks are really important in shaping. And later on, I also introduced a model for creating this kind of directed graphs that promote or hinder any of these 16 motifs that can be 16 3-node motifs that can kind of emerge in these networks. And also in collaboration with Yugoslav Acimovic, we studied how the morphology of neurons affects the connectivity when you place the neurons like on a two-dimensional geometry and then let them randomly connect to each other by the means of their morphology. But then back to this neural network of 150 neurons here. We wanted to speed up the computations in this paper from 2018, and we reduced this Hay model neuron into a kind of a ball and stick model. So the soma is here, and then you have basal dendrites here, and the apical dendrites here as a single line. And we did it in a stepwise manner where we first fit passive and IH properties, and then the calcium and SK channels, and finally the channels responsible for the spiking. And we also argued that this is possible to do, although it has its challenges, but it should be possible to do also experimentally, and that could give us better models in the future that we really know that certain features in the neuron dynamics are due to this and this ion channels. Because as you showed, as I showed in the previous, in one of the previous slides, the parameter variation was really high in this, and they can have a kind of degeneracy that certain behavior can be due to one conductance or another conductance, and we cannot be sure which one it is. And in this paper, we showed that if we reduce this morphology and fit all this conductances along this reduced morphology, we could produce the same network dynamics as was in this Hay model from 2015, and it was up to 20 times faster to simulate. And then when we want to model the EEG signals, what you in general need to do is to solve this this partial differential equation where you have the current sources here and the extracellular potential here, and then the conductivity here. But there are shortcuts how to do it. So one that we and our collaborators have used is this model of 4 concentric spheres. So you model the brain as just a ball, and the cerebrospinal fluid and scalp around it also as layers of ball. And then whenever you place a dipole moment inside this inside or outside of this edge, you can model the local field potential or the field potential. So when it's inside the brain, you call it local field potential, and when it's outside, it's EEG. And they introduced this equation that look on one side a bit overwhelming, but on the other hand, it's a closed form prediction of this model. And so the beauty of it is that you can, when you have the dipole moment, for example, as a time series, you can do it offline, or as a post-processing these EEG signals prediction. So you don't have to really solve this when you have a simple geometry as this. But it has been criticized because obviously the head is not a ball. So there's this New York head model that is also very useful. And here, in principle, you should simulate this, solve this partial differential equation. But they have done it for you, so they have a kind of a huge lookup table that if you have a certain dipole moment here, then and the record here, you get the EEG component. Yeah. By the way, if you have any questions at any point, please feel free to ask. This is a long talk, so let's not necessarily wait until the end. I'd like to next go through this this paper from 2019, where we implemented these methods and tried to explore that what if we change the neuron function at the genetic level? What can we expect to find at the EEG level? And we focus on delta oscillations. Delta oscillations alterations are quite robust phenomenon in schizophrenia patients, but they also it's quite varied. So some studies show that it's increased and some that is decreased, and that's it's likely to be dependent on the brain area where you measure and maybe over it. And we use the same model of 150 reduced morphology Hay model neurons, and the same background synaptic activation and intranetwork synapses. And yeah, we calculate the transmembrane currents for each of these neurons for each time instant, and that gives us EEG at a given distance. Yeah, and then we compare what we consider to be a schizophrenia-like case, and then wild-type networks, which would be the default model. I can go a little bit more into this. So this paper from 2017 shows many of these genome-wide association study identified genes of schizophrenia. There are many more, but these are the ones that are related to fast and slow neurotransmission. So in other words, ion channel encoding genes, which are shown here, and then also other genes related to plasticity and neuromodulation. So this we used as a kind of starting point for our approach, that we looked for data that how do genetic variants in these affect the ion channel behavior first at the subcellular level, and then we can implement it in our single cell and network models. We didn't have at that point, and we still don't have data for schizophrenia variants, but back then we believed that the schizophrenia variants might actually also be like a gene encoding variants, so that when you have the schizophrenia common variant, that would actually affect the channel behavior, not only its expression. But now it's becoming clear to us that it's mostly expression. But anyway, in this study model, and here we varied the rate of the synaptic background synaptic firing. So given less background synaptic activation, we have less spiking in the network, and given more, we have more. Here's just the firing rate over time in this default network. So here you have the rate factor R on the x-axis, and then the firing rate of a average neuron in the network on the y-axis. And in the middle you have the wild-type network, and then these other colors are different versions of ion channel variant, where we change the voltage dependence or kinetics of this calcium channels that are encoded, for example, by this kind of ANSI. And we did it for many of these schizophrenia-associated genes. And finally, we did an analysis where we combined these effects and showed that the combined effects can be large, even though the singular effects are small, which is kind of obvious, but we wanted to show it anyway. We also studied how the network resonates to different oscillatory inputs. So here we had just Poisson background activation, but here we used oscillatory Poisson activation of the that lets us to study that how does the neuron resonate, or the neural network resonate to different frequencies. And we found that this network of layer 5 pyramidal cells quite strongly resonates to delta frequency inputs, and also these genetic variants that we introduced, they at least on absolute terms, they most strongly affected this. So yeah, these results were still done without this EEG component. So I'd like to first go a little bit step by step. How do we get the EEG component? So here's the spike train of the 150 neurons, and here's a membrane potential of a single neuron there. And here we calculate just the sum of all membrane currents. But if we multiply the transmembrane currents with the position vector, so then we get actually the dipole moments. And here I show the y-axis component of the dipole moments, which is the strong. In a single neuron, it looks like this, and in the full network like this. And then the EEG component is actually just a scaled version of this dipole moment. And yeah, here we showed that if we plot it on log-log axis, it reproduces some features that I have shown in reported in awake mammals, cats, if I remember right. But the interesting thing here is that when we take this EEG, the power spectrum of this EEG data here, well, first we see that it's still quite high at the delta frequency range. But interestingly, these schizophrenia variants, they mostly, if not exclusively, affect this activity at the delta frequency range. And that is kind of promising for a few reasons, because delta oscillations, it has been shown that that they originate either from the thalamus or the fifth layer of neocortex. So that is where the sum of the layer 5 pyramidal cells that we model are. And so if the delta oscillations that are reported to be amplified or lowered in schizophrenia, if they are the ones that originate in the fifth layer of the cortex, then this kind of variant should have a direct effect on what is measured in the population differences between schizophrenia and healthy controls. But also, if they originate from the thalamus, we know that layer 5 pyramidal cells kind of post-process these thalamic inputs, and if there's a highly kind of oscillatory activity in the delta range in the thalamus, then it should reflect also on the inputs and output of this layer 5 pyramidal cells, and then it's the layer 5 pyramidal cells are in a way in a position to strengthen this or mediate this EEG signal, and maybe also the differences that are seen between schizophrenia patients and healthy controls. As a conclusion of this first part of the talk, just to recap, so we can model single neurons and networks of neurons and forward model the EEG, and it's not if we restrict to small networks such as 150 neurons, it's not computationally too demanding. And when it comes to schizophrenia, so we showed that manipulations of these schizophrenia-associated ion channel genes, it affects the excitability and the way it resonates to delta frequency stimulation, and that these effects can be observed at the EEG levels. Any questions at this point or comments? Or I move to the subcellular parts. Yeah. So so why model intracellular signaling cascades? Especially if you're interested in plasticity, because in a way many, if not all, aspects that we are interested in neuron dynamics at the millisecond and even second scale, they can be modeled with these conventional models that I showed before. But then when you want to know that what is really causing these slower time scale phenomena in neurons, such as plasticity, we need to understand the intracellular signaling cascades underlying it. And also for schizophrenia, which is my main topic, so as I showed in the figure, there are many of these aspects of slow neurotransmission likely to be affected in schizophrenia, just basing on the G losses. And what we use are the mass action-based models. So we model the evolution of each molecular species that we are interested in. For that, we need the initial concentrations of each species and then the reactions between them, so forward and backward rates. And we can do it either stochastically or deterministically. And here's a figure from a paper from 4 years ago, where we created a kind of a unified model for postsynaptic plasticity in the neocortex. And here we introduced 3 different pathways. We have the PKA pathway, which needs calcium input and cyclic AMP producing inputs for, so for example, beta adrenergic receptor activation. And that leads to PKA activity, and PKA phosphorylates AMPA type 1 receptors, so that it's more likely to be inserted into the membrane. Then we have a PKC pathway, which needs calcium and GQ protein activation, and that can be achieved either by, for example, or acetylcholinergic M1 receptor activity. And that leads to PKC activation, which can phosphorylate GLUR1, so AMPA receptor type 2 receptor. And that leads to endocytosis of this receptor. And then we have CAMK2, which also phosphorylates GLUR1 receptors, and that increases its single channel conductance. Just to give an idea of the model, so there are like 70 different reactions and 20 different non-zero initial costs. And as I mentioned, it was calcium that is needed in almost all these pathways, or all these 3 pathways to be activated. And so here we give the synaptic spine just an input of calcium. You don't see it well here, but there are actually 4 high-frequency trains of calcium inputs. Here it's zoomed in. So starting at 40 seconds, there are 100 inputs of calcium, and that leads to calcium accumulation. And then there's a silent period, and then and that leads to activation of the calmodulin, or first binding by 2 calcium ions, and then finally by 2 more calcium ions. And that is enough for the activation of this calmodulin. And when calmodulin is activated, it activates CAMK2, and yeah, that stays active for a long time. And that is kind of underlying part of this synaptic potentiation. And here, when CAMK2 is activated, it phosphorylates this serine 831 of the GLUR1 receptor, and so it becomes more conducting, more current conducting than otherwise. And then there's also, I haven't plotted it here, but there's also a concurrent PKA and serine 845 phosphorylation, which leads to exocytosis of this receptor. But anyway, all in all, with this model, we can then describe the evolution of the maximal conductance of the synapse in response to this whatever stimulation protocol we give, and then how it either is maintained or slowly decays back to a baseline. And this can be coupled to the kind of models that I showed before. So here we have done it offline. So in this example, we gave just square pulse currents of calcium. But in a more realistic setting, you should take the calcium input from, for example, simulation of in this case layer 2-3 pyramidal cells. So here we could model that how is the calcium influx into the synapse affected by, for example, temporal difference between somatic and synaptic activation of this glutamatergic synapses, as in this STDP. So here I'm showing 3 different time point interstimulus intervals. So when the postsynaptic activation comes before presynaptic activation, then at least here in the time 0, there's less calcium inputs. But when they come more or less at the same time, then we have a stronger calcium inputs, and that leads to then stronger accumulation of calcium in the pre-post versus the post-pre protocols. And that actually then we showed that it well, this is based on experimental data from Sio et al. In a pathway from layer 4 to layer 2-3 pyramidal cells, and they showed that it's neuromodulator gated STDP rule, and here we can reproduce that with our model. So then back to schizophrenia. So I showed this figure earlier, highlighted the ion channels that we could directly use in our models. But there are also many plasticity-regulating genes that we can now directly plug into our model and see that if we change, for example, the expression of one of these genes, how does the response to plasticity-inducing stimuli change? And in this submitted paper, we have studied a particular protocol used in this EEG phenotype that I mentioned, the LTP-like plasticity of the visual. But here we are now using rather than using theoretical guesses that maybe these functions of these proteins are changed in the same way as some random variants from the literature. We now have RNA expression data from patients versus controls, and that we can directly use as a kind of a guess that how are the genetic what is kind of underlying these differences in the behavior of these proteins. So we rely on this RNA expression data. It's been measured from anterior cingulate cortex and prefrontal cortex post-mortem. So here we first did a theoretical study that if we just change the initial concentration of any of these proteins by plus minus 20, we get either large or small effects in the plasticity outcome. Here we then did a combination of where the alteration of each of these proteins was directly taken from this RNA expression data. And here we see that if we take the alteration from anterior cingulate cortex, we get a mild but still observable difference in the LTP outcome. While in PFC, you can see that there's a difference, but it seems very small. I should, by the way, explain what is here on the x and y axis. So this is still a conventional LTP, so it's not yet this BP protocol. Sorry for the misleading title. So you have a frequency on the x-axis. So when the stimulating frequency is below 10 Hertz, then we get LTD depression. But when it's more than 10 Hertz, then we get LTP. So this is a kind of a typical BCM-like plasticity. And so to justify that it's still meaningful to look at these small differences, we did a subject-wise analysis. So instead of taking group averages that, for example, PP1 concentration is a certain percentage less in schizophrenia than healthy controls, we do it subject by subject and then simulate the plasticity curve for each of them, and then do the statistical tests afterwards. And we find that in ACC indeed is quite strongly. For PFC only in some stimulating frequencies. And we did the same for STDP. Maybe I'll skip most of this in the interest of time. And the same result. But now coming back to this VEP-like plasticity, the LTP-like plasticity of VEP. So there they used 10-minute intervention block. So for 10 minutes they showed the subjects the reversing checkerboard stimuli. And so we model it so that for each of the reversal you get a calcium input. But we study both GluR1 dominated and GluR2 dominated synapses because we don't know if the plasticity that is seen is LTD or LTP. And we do the same as I showed before. And here the combination of this, based on the RNA expression data, is a bit stronger than before, but still the same trend that in PFC the change is small, but in ACC it's a larger one. And also the statistical difference was stronger in ACC. And if it's GluR1 dominated, if it's GluR2 dominated and LTD, then we see little or actually no statistical significance at all. Using this RNA expression data, we are now also doing this delta oscillation analysis again. And so Jan Fredrik has been working on this and has been able to show that it actually seems to be reduced the delta oscillation power, both when using PFC and ACC data on ion channel expression of these schizophrenia-associated genes. So that's kind of surprising for us because we believe that the increased delta oscillation power is a more robust phenomenon than the decrease. Finally, we wanted to validate our model predictions. Our collaborators have indeed collected this data from hundreds of patients and healthy controls. And we wanted to see if there is something in the polygenic risk score of schizophrenia in these participants that could explain this strength of this modulation or LTP-like plasticity of the VEP. And we focused on those genes that we believe to be involved in postsynaptic plasticity. So here you see a baseline. So before there's any intervention or any intervention, the baseline ERP looks like this for the checkerboard reversal. There's the C1 peak, B1 peak, and N1 peak. And then they get modulated after this intervention block by some 2 to 3 microvolts. And we then calculated the correlation between these polygenic risk scores and the modulation amplitude. And we found that the polygenic risk scores that were based on the SNPs found in genes regulating plasticity or encoding ion channels was statistically significant. And likewise, those plasticity genes that only had an effect on the PKA, or mostly on the PKA pathway. And also if we only concentrate on ion channels. But if we only consider the PKC, there was actually opposite effect, but it's non-significant. So in conclusions, so many schizophrenia and plasticity-associated genes and proteins affect the LTP and LTD in neocortical synapses. And if we change the RNA expression in our model, if we change the protein expression in our model corresponding to this RNA expression of schizophrenia versus healthy controls, we find decreased LTP amplitude and decreased excitability of the pyramidal cells. And yeah, the LTP, especially when we consider this protocol used in this VEP modulation, we found in our models, and the association between this VEP modulation and PRS is kind of supported. That is fine. So I'd like to finish with some what we believe to be the future challenges and opportunities. So the modern models are good, but they lack genetic detail. So there are many different RNA encoding genes for especially potassium channels, but also other channels. And experimental data should be used under pharmacological blockages and maybe even genetic manipulations to get better and better models. And yeah, the models of endophenotypes are at the most as good as the network models we have for these are difficult to tune, as many of you know. Yeah, and now there are coming more and more RNA expression data, but ultimately we should also need protein expression data because that's what our model is based on. Yeah, there are larger samples of clinical genetic data becoming available. And then these induced pluripotent stem cells could work as a bridge towards personalized medicine and personalized physiological modeling. So we already showed that we can tune our models subject-wise and then calculate, for example, group differences based on those. But the same could be done using these induced pluripotent stem cells, and yeah, that could have a range of opportunities. Yeah, I'd like to conclude here. Yeah, please, so I'd like to conclude here, and thanks for your attention. Yep.
About the pyramidal neurons, at least in the first experiments, were in vivo patients or post-mortem?
So the endophenotypes of for example, increased delta oscillation, you mean? So those are the electrophysiological data are always from alive patients. So, but then genetic data, you can this RNA expression data you can get either from blood sample, which is considered not so good because we don't know how it correlates with the brain expression in the brain. But you could, and then there's post-mortem RNA expression in the brain. So those are the two main sources that we have used. That's also where iPSCs could help. So if it becomes more cost-effective to take these skin fibroblasts and make, for example, cortical organoids, then you can do expression analysis on those rather than.
Does it explain that the motor cortex is the M1 area? There's those fifth layers, pyramidal neurons. Is the primary motor cortex, or which area are they measured? How they correlate with schizophrenic patients' symptoms? Like, it's not more likely to be temporary, like when they have auditory hallucinations or other areas than the motor cortex.
Yeah, I wish I could answer that question, but I'm not an expert in when it comes to brain area differences. So the models we use, they are tuned to somatosensory cortex and actually rat brains. So eventually one should have a brain area specific models in humans, but but also the clinical phenotypes of increased or decreased oscillations. So that is dependent on the brain area, but also it's not very well yet known. So it could be that they measure here in this electrode significant difference. But it may be that the source is somewhere else than actually closest to the electrode because of the folding of the brain and
so little distance. Oh.
Yeah, so this plasticity model is of course impressive. I was wondering how well does it is it assumed that this is the model for all glutamate synapses on spines in all layers in the cortex, or what is known how different this is, or this is on the basal dendrites, right? It's
yeah,
the dendrites. Oh.
Yeah, we aim to make it as a kind of a generic model for neocortical synapses. There are differences. So also in this paper from 2020, we after creating the generic model, we retuned it to plasticity data from different cortical areas. We have ACC, PFC, auditory cortex, visual cortex, and entorhinal cortex, if I remember right. And but then again, the literature data that we use for those, they didn't all specify that what if you made this experiment in presence of a certain blocker, PKC or PKA pathway blocker. So then when we fit our model to those data, we cannot be sure that do we have all those PKA and PKC CAMK2 pathways correctly tuned. We just know that our model reproduces those data.
If there is no question, I can ask one last maybe. In your plasticity model, there was no changes between when you lose this protein below 10 hertz. Do you have any intuition why is that?
Right. So this figures you mean?
Yeah.
Well, one is just visual. So if you zoomed in, for example, here you might see quite radical changes. But yeah, that was a kind of also the model was tuned to maybe so that it maybe doesn't produce as large depression as it could. Although when you look into the data, you maybe don't find either that large depression as we found So, but yeah, then why is this? Was this less significant? For example, here, I haven't studied it, but also the set of genes that were associated with the gene with schizophrenia seem to be more in the PKA pathway than PKC. Why our model shows that it's more significant, the differences in LTP than in LTD.
No, thanks.